we'd like to invite Rice Technologies to the stage.
Good morning. My name is David Rice. Has developed a multi-purpose contamination sensor. This is patent pending. Here,
I am Marcia Silva. I'm a scientist with the university, and my laboratory is specialized in material sciences and water technology. Such a pleasure to be here this morning with you. Such an honor.
our offices are in the same building, the Global Water Center. This building has been specifically designed to aid collaboration between large businesses all the way down to startups. Not only that, the university's research laboratory is also in this building, and that is also entirely open to the company. What's in this glass of water? Can you see anything? But you can't tell if it's contaminated or not. But we have developed a device that actually can measure whether there's any contamination in that water. Not only that, it can do it in real time.
I have 2 children, and when I take them to the beach, I really wanted to assure that they are safe. So I'm wondering if they might have a skin rash or diarrhea. So I'm wondering about the amount of bacteria that is in that water. Do you know how these analyses are performed? So currently, researchers follow APA method 1603, where a sample of water is collected. It is filtered using a membrane filtration technique. Sample is placed in an incubator and left there for 18 to 24 hours until we can count the amount of bacteria in that sample. So that means when we look at the beach signs, they really reflect the yesterday's conditions of that water. What if we could have a sensor that could detect bacteria in real time? And how about the case in Flint, Michigan? Do you remember in 2016 when a large concentration of lead was detected in drinking water? At that time, the major concern was with the children. So a concentration of lead, high concentration of lead for children may cause damage in their kidneys, their nervous system, may delay their development. What if we could have a sensor that could detect lead in real time? And how about medical application where a patient who depends on dialysis bag and the quality of the water that is in the dialysis bag may interfere on the health of that patient? Do you know how many analyses have to be performed in that dialysis bag? They are listed here, but I can guarantee you cannot even read it. They are too many. What if we could have a sensor that was able to detect all these compounds in a single shot?
In the world, there are nearly 200 semiconductor foundries. They all use lots of pure water in many stages in the production cycle. Currently, they take, the way this is done, they take city water, they filter it and disinfect it, and then use the water. To check whether that water is working well, is good, they use a device called a Conductance Meter, which are excellent devices, except for one thing. They can't detect if you've got any biological contamination. So if something goes wrong with their filtration system, boom. They've got to shut down the system. They They've got to clean it out. They've got to throw away all the batch that has been destroyed, they've been ruined. Clean it, start it up again. These buildings, these fabrications buildings, typically run about $6 million an hour. It's enormously expensive to shut them down. What if we had a sensor that could detect this in real time? Throughout the world, aquaculture, growing fish, is a major business. It's a $200 billion business throughout the world. Because of the sheer amount of water they use, they can't just use water, they have to recycle it. And what they do, they filter it, disinfect it, same process, and then put it back into the fish. If they use the same conductance meters as semiconductor factories, if they have, and they do, this happens, if they have a biological contamination, you'll start to poison the fish. Now, unlike a foundry, if you kill the fish, you can't just replace them, you've got to grow them. So you could kill months of product, possibly even a year's worth of product. And that is hideous. What if we had a sensor that could detect it in real time? This is a picture from the International Space Station. Because of the weight of water, they can't continuously supply water to the space station. They have to recycle it. Unfortunately, the recycling equipment is very complex, very prone to failure. There's a lot of maintenance required. The reason I'm in this building today is because a proposal I sent to NASA was use our sensor in multiple places throughout this unit, and it will do two things. One, it will shut the system down immediately if anything goes wrong, and two, it'll tell you which particular module has failed. So it improves maintenance and doesn't allow and other failures continuing on down the line. This is this will fix a real pressing need for NASA. There are many sensors on the market, but their limitations are they tend to work over very narrow ranges. They can't, if they can detect very low levels of contamination, they can't detect high levels. If they detect high levels, they can't detect low levels. They're also generally very specific. You can get a meter that's designed for suspended solids. Works excellent, but it can't detect dissolved solids. That's what a conductance meter does. You can get one that detects ammonia, like they use for fish, and so on and so forth. They're very nasty. They're also remarkably expensive. Many of them are not very fast in their response. They take a long time to go. If you draw a curve, this is the sort of thing you'd see from a regular meter. It'll detect over a narrow range. The two grayed out parts in this at the ends you can't measure. That's the high end and the low end. But you can detect in the middle. It'll do 1,000 to 1,000,000 range. That's very common. Next. This is what our meter does. This will measure, but we're using E. coli for this one just because it was easy. We can measure down to a few bacteria, this is between 1 and 10 per milliliter, all the way up to 100 million bacteria per milliliter. That's enormous range that can be measured. Next.
Unlike the other sensors, rice sensor has the capability of detecting organic compounds such as bacteria and viruses. and inorganic compounds such as phosphorus, ammonia, and salts. It works in real time. It's a single device. It's very selective, sensitive. There's just a need for a small sample of water. No specialized personnel to operate the equipment, very low cost, low energy consumption, and it's portable. Rice sensor is one solution to all the environmental, medical, and a variety of industrial applications we have presented here. Rice sensor, here we brought one of the sensors we have, and we will pass it around for the judges right after our presentation. So it's this one that we fabricate in the lab. It has this size, but we can fabricate it smaller or larger. We can actually roll it and place it inside of a pipe for detection. There's and how can you use the sensor, right? So it can be a simple adding a drop of water inside of the sensor in the sensing area, or it may be placed in a dynamic system when there's flowing water. Well, this is a fun picture at the school of my children. It was a science day, and we took the sensors there. So we had about 100 children from K4 to 8th grade literally squeezing the sensor for about 1 hour. And the sensor survived. I think that's a very nice way of testing how robust the sensor is.
Okay, what this says on the left, this is a graph against time of contamination. It's a single flask, and we had 2 sensors in the same graph. It's just distilled water. So you start off with 0. A little bit changes over a day. First 24 hours, that's what happens. Then just sprinkle really just a few grains of salt in it. And you go, whoa, we've got a massive change in contamination. On the right-hand graph, you can see there actually is 2 traces. But what you also see is that both sensors track each other exactly. The contamination is changing. The dynamics of the contamination is, I don't know, but it's changing subtly. But the 2 sensors track and show exactly the same results. That's repeatability.
Just as a recap, there's definitely market match for the sensor. So applicabilities in the medical field, environmental field, semiconductor, pharmaceutical industries, cosmetics, aquaculture, NASA, definitely Department of Defense as well. And why not smart homes? So those are the features that the technology already has. Why not add the water quality icon? Dave? Cool.
Okay, the next steps. As we've got to partner with, find a partner that can help us, can get to the market-ready product. This partner will also have to have access to electronic assembly and test, and hopefully, well, essentially, some established marketing routes. Cool. To complete this, what we're asking for from them is 450,000, and which we'll be selling them within a year. We know every single step to do this. There's nothing that's not unknown, and we are that close to getting there.
And this is the market waiting for you. So the global market size of sensors is over 95 billion. Combined market of biosensors and chemical sensors is about 20% of that. So we are talking about 19 billion. This sensor is a game changer, and you know that. So it will change the way people do things in a variety of fields, environmental, medical field. This is extremely exciting to us. It couldn't represent better what NASA iTech represents or was looking for. Technologies that can solve problems here on Earth and that can easily be used for solving problems in space. Dave.
Okay, this is just the two of us, but because of where we have our offices and who we work with, we have access to people with electronic knowledge, mechanical engineers, biologists, molecular biologists, IT, legal people, and attorneys. That is built into where we work.
This technology lines beautifully with strategic trusts presented early, mainly ST5, to enable humans to live and explore in space and in planetary surfaces. That water is definitely life, and we need rice sensor. Thank you so much for your time today. Thank you for your attention, and we are now open for questions.
I'll give these out.
Thank you for your presentation. I have a question talking about the technical composition of your sensor. So going back to your competition, I do know of a lot of sensors in this space, and you had mentioned that those sensors have limitations in terms of what they're able to detect. So I'd like you to be able to touch upon what technically separates you from the competition. So for example, I know that in other sensors, they'll run liquid through a microfluidic channel lined with electrodes. So what is it about yours that has the technological superiority to be able to test for more?
The way it works is you have two sets of electrodes. They're like combs, and they interlock with each other. They're insulated from each other, and they're insulated from the water. One puts a charge into the water, and the other one observes how the charge discharges. That discharge is critically dependent on what's in the water. And we've learned how to understand this. That is the basics of how this works.
The major change here is also the ability, first the incredible dynamic range of a sensibility range of this sensor. Also the capability of working with inorganic and organic compounds. It's so sensitive that it's capable of differentiating types of water, and we did learn that by coincidence in the lab. So when we have ultra pure water, or for those of you who have the scientific background, or if you are have an ultra pure water or the deionized water or distilled water, they are all absolutely clean water, but yes, they do have a huge difference in composition, and the sensor was able to detect the differences in these types of water.
Very exciting product with a lot of potential. I'm very
Absolutely.
enthused for you. I'm curious, you won a big award, a fairly nice little award, $50,000 in 2013. What have you done to this point? What's taken so long?
That's a award actually. A different product. It was not for this. Not the same product. No. And what that was is it doesn't matter. It's a different product, and that went on successfully. We sold $600,000 worth of equipment, but I had a falling out with my partner. The whole thing died. But I was now in this building, and I couldn't leave it. It was such an exciting place to be. And that is where this was, the system net was then developed.
Yeah, and now it fits nicely to mention how we met. So in this very nurturing environment we call the Global Water Center, where there's a variety of startups and water industries and also universities. So Dave Rice had started his company in 2013 with that one project. And he had this non-invasive technology for detection of leaking pipes. When he stopped in my office or in my laboratories on the 7th floor of that building to deliver a misdelivered mail, that's when we started talking. I was writing on a proposal for detection of suspended solids in sludge, and we brainstormed we came up with the idea of pushing this more to my interest in that case for the water technology applications. And we start working in 2014 together. So it's pretty much almost 4 years working on this technology to take it to the level that is right now.
What happened, I was working on this laboratory bench, and the bench next to me, there was some students developing a device to measure bacteria in water. They had some doctors over to look at what they were doing, and I was just sat there, ooh, this is interesting. After they'd gone, there was just Marcia and I, and we said, what happens if we put some of their samples of small quantities of bacteria in water on our sensor? So we did. It was fantastic. It was so sensitive. It just blew our minds away. So we made a major pivot, and now we said, forget the sludge bit. We're working on this.
And then because we thought that after realizing that the sensitivity of the sensor for different types of water, we believed that it could work for viruses. So working with my collaborator at the Public Health in Milwaukee, we took the sensor to his lab, and I gave him a call, and I asked, we do need a sample of viruses. And he said, oh yes, I have the viruses for you, just bring the sensor. We took the sensors there, we tested, we couldn't even think about it. It was so exciting. So he took the sensors there with one of our students, and we tested the sensor for 2 different types of viruses. And we were just laughing at each other. It was just incredible, too much exciting to realize that the sensor was able to deliver 2 different types of signature for the 2 types of viruses we were testing.
Couple of questions. Can this be used in any other kinds of solutions other than water?
Well, yes. Sorry.
And they're not necessarily fully related. And have you imagined the impact of the environment of space and any impacts that may have on this? And how do you clean this to maintain that type of sensitivity? Because it sounds like with that sensitivity, you have to have wonderful cleaning.
Yes, you do. We know that we're working on a shoestring, so we haven't tested everything. But when we did the virus test, the viruses are not in water, they're in another fluid. No, I don't really know what it is. Do you know what fluid they're in? The viruses. It's not water. No matter. It is another fluid other than water. So we know that it works in other fluids. What was your next question?
About the space environment.
Okay. The electronics are just simple electronics, so you can just do that. That'll work. The sensor itself, at the moment, consists of fiberglass board, copper conductors, and epoxy insulation. If you change the epoxy to glass, which is what we're expecting to do for the final device, there's nothing to degrade. That's going to work in space.
I really think you are talking about how many more applications. We did bring to this group here the applications for water, and primarily being a facility, the Water Technology Accelerator. Every time we conceive ideas, we are really thinking about water applications. But very often we do find applications in the medical field, and very often in some of the other technology we have, people ask, can this be applied for other fluids such as air? The answer is absolutely yes. That's not how the technology was conceived, but transferring to another application is quite an easy task. In terms of other fluids, you were in my earlier version of my presentation, I gave a little more taste about the application for viruses detection. And for that particular case, other body fluids such as saliva or semen or urine could definitely be great candidates for this sensor.
And the last was about your protocols for cleaning.
Oh, cleaning, yeah. That actually was, we struggled at the beginning because we didn't really appreciate the fact that if you're trying to measure very low levels of contamination, you've got to have a completely sterile sensor. And then you go ding, I was dumb. But the cleaning turns out to be so simple. You spray it with alcohol, then a couple of washes with distilled water. Boom, it works. That's all we've been doing, and it works excellently. It's not complex.
Is this a conductivity measure? Is what you're measuring? Is electrical conductivity?
No, it's actually no. It measures how the charge changes in the water. You put electrical charge, you put so many billion electrons in the water, and you just watch how they decay.
So how do you get selectivity?
Selectivity is that's a different thing.
Right. Right now, sensor is in phase 1. In one of the stages that slides that Dave showed you.
We haven't got it.
So we are literally 12 months from having this product on the market. That is for phase 1. Phase 1, the sensor is extremely sensitive with all the properties we have showed you. However, that is when you have a sample with that one compound of interest. So once you have a variety of compounds together, which would be, let me see if I can find that slide. It's meaningful at this time, so let me see if I can find it. Right here. Okay, if you have another application that the sensor is ready right now, is this one. Dialysis bag. So one of our collaborators from Medical College of Wisconsin, they came to our lab and they asked, can this sensor be used for dialysis bag evaluation? Where in that particular case, they are not interested to know how much of each of these compounds they have there. They only want to know it's a yes, no test. That's all it is. Can I use in the patient or I cannot use in the patient? The sensor is ready for this. If you are talking about, I do want it to detect specifically E. coli or Legionella in water, then we are talking about phase 2 of this project. That's where we are going. My lab is specialized in material sciences, biotechnology, and water technology. So my lab has expertise to modify the surface and make it specific for target-giving compounds. And by the way, I do have other technologies in my lab, not sensor technologies, but filtration materials that have already that capability. So it's quite simple to me to transfer that technology to sensors.
Basically, if you viruses work by having the right shape and it locks into a part of the cell. That's how they work, the shape. If you etch the surface of a glass like you do in semiconductor manufacturing, that's the process you're going to be using, and you make just the right shape to accept just that one virus, only that virus will adhere to the surface. The rest are going to be actively repelled. So in a matrix, which is what you're going to have nearly all the time, you can tune the surface to one particular bacteria or virus.
So you have identified many different applications and many different markets for this sensor. Have you been able to talk to these different markets to see if they're really willing to incorporate the sensor and have a better reading than what they already have, like is good enough what they have?
I want to tell you this funny story. When I called my collaborator at the public health, and I called him and I said, hey, I need a sample of viruses. Why do you need sample of viruses? I just wanted to test the sensor for detection. And then he stopped me and said, hey, I don't know of a sensor that detects viruses. And he kept talking, so it was over the phone. And I said, hey, stop. I know about the sensor part. All I need is the virus sample. He said, where did you find the sensor? We are developing the lab. He said, you have to tell me when this is ready. We need this in our lab. That was one instance that he got absolutely, they do services for the community, right? So they receive thousands of samples per day. He was he was mesmerized by that. That was one instance, and the other one was when we received my collaborators from the Medical College of Wisconsin in my lab. Actually, I did bring them, not for this one sensor. I brought them to I have other sensor projects in my lab, and that was an optically based sensor that was in one of the benches. I brought them to see that one application. However, the benches in the lab, right? So there was one application in one bench, and right across there was the rice sensor application. So I brought them there to see the number one application, the optical based, and then they listened to everything, and then they turned to rice sensor. They said, hey, can't we use this one here for our samples? So they were more interested in this one application than the one that I was primarily interested in showing them. So for a variety of reasons, it was just ready to do what they were looking for, which is the slide that's displaying right now. A yes, no test. Or another application they mentioned for urine analysis, and I am not a physician, but learning from them, they are interested, there's a threshold of certain cells that are interested in urine. They don't care necessarily how much is there. They wanted to know there's a threshold, is it above or below? And our sensor has the capability to do that right now.
Time. Thank you very much.
Thank you.